have a number of speakers. Yeah, and we have a number of speakers lined up. So I'm just going to pass this to Roslo, who will give us a recap of the meeting yesterday, and then Rose will also introduce the chair of the session for today. So Rose, over to you, please.
Yeah, hi, welcome everyone again. As thank God said, this is day 2. Yesterday was basic biology, and I just want to take this opportunity to once again thank everyone who spoke yesterday, but also especially to Michael Ginger from the University of Huddersfield, who chaired not one, but both of our sessions yesterday. Yesterday we had a keynote speaker from Jonas Korlach from Pacific Biosciences, who gave us a really exciting overview of kind of all the new technologies that we can start to bring to bear against euglenoids and their very interesting genomes. We also had 2 invited speakers, Masami Nakazawa and Pika Euglenoids, who spoke about anaerobic mitochondria in euglenoids and euglenoids for One Health, respectively. All of these sessions were recorded, and if you didn't get a chance to get to yesterday's session, I highly encourage you to go and have a look at the recordings once they're made available at the very end of the conference. We also had then an array of short talks from a variety of people that ended in a really engaging discussion, and I hope that we can replicate that today. As thank God said, today's theme, if you like, is Ecology and Environment, which is going to be chaired by Kevin Tyler from the University of East Anglia. So without me rabbiting on much more, I'm going to pass straight over to Kevin. Over to you.
So, do we have Kevin here? Okay, I'm not sure he has joined. So I'm going to chair the session then in the absence of Kevin. Is Kevin here? Okay, I think I'm going to chair the session. So the first talk today is Professor Edith Hurd. Edith is the Director General of the European Molecular Biology Laboratory, EMBL, and she'll be talking to us about the new EMBL program and how this connects to the Sustainable Development Goals and other microbial eukaryotes. So, Professor Head, could you please share your screen when you may? Yeah.
Hopefully this will work. Okay, I've lost my screen. That's not good. Sorry.
Yeah, I think it's just at the bottom. There's a share screen.
No, yes, I just couldn't find my PowerPoint.
Okay, yeah.
So now I have it, and hopefully this is okay.
Yeah, that's fine.
Okay,
let's go ahead when you. Yeah.
So thank you so much, thank God, and thank all of you for being here and for inviting me. I do feel like a little bit of an outsider. I'm not a euglena specialist, but I'm fascinated by symbiosis and by life out there. So I'd love to know more and learn more. And as the Director General of the EMBL, this is a special time, a unique time actually, an opportunity for an organization like EMBL to reach out to communities of scientists working on topics such as understanding the diversity of life in its natural context. And I think that this particular topic of euglenoids is highly relevant. So I hope that I will at least tickle your interest about the future at EMBL. So in a nutshell, EMBL is actually Europe's only intergovernmental organization for life science research. We have to deliver on several missions. We have to perform research. We have to deliver access to infrastructures and scientific services, including at the European Bioinformatics Institute. That's one of our 6 sites. We also deliver services through access to synchrotrons, for example, for structural biologists, and imaging, a new imaging center that has just opened up on our Heidelberg site. We also have to provide training of the next generation at all levels, from masters through to group leaders and postdocs, as well as engineers, technology developers. We also have to do innovation and translation. So much of the research and technology development that gets done at EMBL leads to innovation and translation. And last but not least, we're here to try and integrate life science research across Europe. But I should say that we have an even more global role, as I'm sure, thank God, knows. Through some of our sites, such as EBI, we reach out in a very global way. So we're actually a very diverse, multi-site, multidisciplinary organization. We have to serve 27 member states who support us, and we have to provide access, as I said, to many different types of services. And for example, EBI data services have 82 million daily web requests a day. And our experimental services have almost 7,000 annual user visits. Oops, sorry. So in my next slide, I just want to say that research on biodiversity has been going strong actually for many years. More than 10 years ago, there was a big project that was set up called Tara Oceans, and this was actually driven by Eric Kaszecki, who was actually a head of a unit in EMBL Heidelberg, who set out to try and explore oceans at the molecular level. And this project has led to, which was extremely collaborative and very global, has led to amazing discoveries, including thousands of viruses, bacteria, and other organisms, and many of which are actually crucial to our planet's health. And this has provided, really important data on biodiversity in oceans, both at the genomic level, but also now using imaging at a sort of whole organism level. And some beautiful stories of symbiotes have come out of Tara Oceans. We also have research. Actually, I'm sitting in EMBL Grenoble today. So, for example, there are groups working on trypanosomes, and there's a big interest to try and understand nature versus nurture, or the interaction between genomes and their environment to give rise to phenotypes. And this is the Nematostella sea anemone, which responds to food levels by growing different numbers of tentacles. So we also have a lot of services that enable biodiversity research. So I've already mentioned EBI, and in terms of genome annotation, EBI provides the infrastructure and the resources for many, many large-scale genome annotations and initiatives, including, of course, the Earth Biogenome Project, and Darwin Tree of Life, and others. EMBL was also behind the partnership with Google's DeepMind to provide access to AlphaFold, the revolutionary database that allows you to go in and find the structure of your favorite protein, particularly human proteins, but stretching out to all organisms. And hopefully this is something that will also be of interest to the euglenoid community soon. And last but not least, we are all about open data. EMBL, everything we do is geared towards trying to make sure we share data and tools, of course, as easily and quickly as possible in order to accelerate knowledge and research. And of course, this has an impact on health, and the pandemic has been a beautiful illustration of that. But in looking to the future, also in other areas, including biodiversity research and, of course, agricultural research. So we are very committed to this open science area, and it's something that we try to even lead on. So the new program, which is why, thank God, invited me, is summarized in this one slide. Our vision was to stretch beyond just the molecular biology of life in a test tube. EMBL has traditionally been known to work on molecules from the atomic scale right through to molecules in cells, tissues, whole organisms, but really try and understand life in its natural context, which is in communities and populations, and in highly changing and variable environments in some cases, sometimes man-made environments and insults, and other times natural environments that have to change, or that change and whereby organisms have to adapt. And so it's in this context, trying to understand relationships between organisms and how this takes place, whether it's symbiosis or mutualism or infection or predation. We believe that EMBL can provide tools and resources and expertise that could be useful to many communities of scientists. And so at the heart of all this, of course, is the concept that one needs to understand life in the context of ecosystems, and healthy ecosystems are essential for life on this planet and the well-being of us humans as well. So the new program could be considered in three sort of pillars. One is this bold vision that I already mentioned, to understand life in context, an environmental vision at the molecular scale. The second is to really try and rise up to societal challenges. And I think this is something that we're all keenly aware of because of what we've just gone through, the COVID-19 pandemic, but also looking around us, climate change, biodiversity collapse, etc. And these are challenges that we feel EMBL scientists and our infrastructures can be useful and can lead to both new technologies and innovation. And we want to do this in a highly collaborative way. So in fact, this program is designed to reach out to people around the world, to try and bring people from different areas, for example, including ecologists, environmental scientists, as well as mathematicians and physicists. Theory is part of this program, trying to model ecosystems and trying to model the interactions between organisms in a very detailed way and make predictions. So it's very collaborative and interdisciplinary. And this is, in fact, how we summarize it in a very concrete way. So EMBL is spread across 6 different sites, institutes where the work is done, although we want to spread across Europe and the world even more so. But we build up on what we already have. We have very strong basis in structural biology, computational biology, genomics, developmental biology, etc. And so what we would like in this new program is to move into areas that we're not as well known for. For example, understanding planetary biology, which I'm going to say a few words about now, but also microbial ecosystems, infection biology, and human ecosystems. And we can do this very naturally because we have a high turnover. EMBL has a rule whereby people cannot stay for more than 9 years. So in any 5-year period, which is how long each of our programs lasts, we have almost 50% turnover of group and team leaders. So this is a fantastic way for us to evolve very naturally into new areas and this program. So I want to sort of zoom in on planetary biology very briefly, but because I think this is perhaps one of the most relevant chapters of our new program that could perhaps be of interest to you. So when we say planetary biology, of course, we're just talking about planet Earth. We don't believe that there's much of a planet B. I think planet A is already plenty to be proud of and worried about. So we want to explore ecosystems, first of all, out in the wild, a little bit like Tara Oceans. We want to develop a systematic approach in collaboration with people in many different institutes to try and get a better census of the molecular nature of ecosystems. And one of our flagship projects is going to be coastal ocean and soil sampling that I'll say a few words about in a minute. Another area that we feel we have to dive in is trying to understand communities of organisms in context. So trying to set up experimental studies to validate and sort of get mechanistic understanding of the communities that exist out there and see whether we can understand environmental responses at single cell resolution, for example. We also want to make sure that the genomics that is being done in the world and that could be useful for understanding is done in a proper way, systematically, a little bit like what Tara Oceans do. So systematic sampling and then producing data that can be made sense of using, of course, things like artificial intelligence, but also applying theoretical approaches to try and model what's going on and perhaps provide biomarkers for molecular tipping points, for example, in arid zones. And last but not least, of course, data science is at the heart of everything we do at EMBL. We would like to expand EMBL's data services to enable some of the key research that we do in these areas of both human and planetary health, using approaches such as AI. And I think AlphaFold is a beautiful example of that. So as I already said, the way we will do this is bottom up through our existing institutes and people, and with an extensive collaborative framework that we're setting up right now with population geneticists, climatologists. I should say that EMBL is an observer at COP26 right now. There are several people from EMBL actually in Glasgow. We want to reach out to these communities of scientists so that we can work together. And just in case you're wondering, you know, this is just a lot of words, we thought about this really carefully. For example, in planetary biology, we want to explore, you know, who is there, make repertoires of species, genomes, defense repertoires, try to capture evolution in action, because you can, especially for some of the microbial communities, and then understand who's out there, but what do they do? Try and get a handle on metabolic networks. We already have a lot of science going on in these areas, and the roles of specific organisms in their ecosystem, identify, you know, keynote species, and try to understand better what the impact of changing environments are on such species. And also, how do they actually do what they do out there? You know, functional ecosystems are essential for us to be, well, for us to survive, actually. And so we feel that we as molecular scientists should be able to help bring some of the knowledge that's needed to go out there and provide new biomarkers and novel probes for ecosystem function. So shown here on the right, you can see that we're going to use many different approaches, from imaging to omics, modeling, and across many different timescales. And one of the flagship projects, as I mentioned, is this coastal sampling project, which right now is aimed at European coastlines, but of course it's something that one can imagine going even further, both inland and more global. And here we want to actually create or bring mobile labs with state-of-the-art technologies to areas and sites where organisms of interest or ecosystems of interest are available, with the scientists who have been studying them in some cases for many decades. So we want to try and help, you know, leverage the talent and expertise that already exists, and provide our own expertise in this collaborative way. So this means bringing together teams of field specialists, field scientists, imaging specialists, analysts, and we've already started to do this, very much inspired, as I said, by Tara Oceans, which was this huge and successful project. And in fact, this coastal sampling project will be done in partnership with Tara, but also with these mobile labs and other fleets of vessels. And part of the key to this is not just doing the science that we need to do to get to grasp life in context, but also trying to engage the scientific community in a much broader way. And so we believe that it's actually essential that somehow the communities of scientists and public come together to get a better understanding of what it takes to understand real life. And I'm showing you some beautiful images here of life in context, but we need to do this using the tools that we have for sharing data, for example. And I hope that maybe the kinds of tools and approaches we have could be of use to the global Euglena scientific community. So some of the applications and impacts of what we're doing already, in fact, not just what we're going to do. We're already working, for example, on algal systems to try and develop and scale up carbon fixation solutions when it comes to thinking about climate change. These are projects already ongoing. We want to try and identify those organisms and functional traits that could be of biotechnological relevance. And here again, I know that Euglena would be very interesting from this perspective, given how they've been implicated in CO2 sequestration, and they could be potential candidates for climate change mitigation in these sorts of approaches. Planetary health biomarkers, we have collaborative projects to try and capture, as I said, some of the tipping point markers that we need to have. And last but not least, we need to think about biofuels and microalgae cultures for fuels that could be used, or even for animal feed are actually ongoing at EMBL. And we need to direct some of the technologies we already have, and new ones as well, towards the environmental challenges that are out there. And I'm just showing you some publications or some ongoing work from EMBL to show you that we're deadly serious about this, you know, understanding collaborative or competitive microbial communities. And this is done both communities of microbes in the gut or in soil or in oceans, solving the impact of nanoplastics using structural biology approaches. That's an ongoing project, and actually searching for degrading pesticides with some of the useful microbes that are out there and that we can actually harness to this approach. And these are projects, as I said, that are ongoing at EMBL. So just to finish up then, we hope that this new program will benefit society through through the tools and the resources that we can provide to the scientific community, the research we do, as well as the innovation and translation. And we want to set up a new innovation ecosystem that will bring together stakeholders in different areas, not just academics and industrial partners, but coming up with new public-private partnership opportunities, linking up to some of the areas that we're not traditionally linked up to, for example, agritech, and also in the area of consumer goods. And so we hope that from this program, will help us to rise up to the global challenges that are needed to fulfill the United Nations goals. So some of the most relevant ones for us are shown here. Goal 3, ensuring healthy lives. We also work on cancer, on diseases of the microbiome, on infection. Goal 13, to combat climate change. And here, I think, as I mentioned earlier, that euglenoid organisms would be particularly interesting if you are interested. Goal 14 would be to try and conserve our oceans. And this is obviously relevant to our flagship TREC project. And finally, to protect ecosystems, which is goal 15, and to try and halt the collapse of biodiversity. We need to understand what's happening in order to prevent it. So, the value of our program, I hope you've seen it. This is just a summary. We hope that this will be a real facilitator that will include communities such as your own community to bring together, people from different disciplines to work together, provide tools such as data portals where you can explore your favorite organisms in their ecosystems and bring together different types of data sets. And we want to train the next generation of scientists. And this is something that we're setting up through a number of tools, including new courses, conferences, as well as visitor and sabbatical programs. And last but not least, we would like to influence policies with science and scientific evidence. And so with that, I will end just by giving you an advertisement for another conference, which will be happening later on this year on the 3rd of December, which is the 22nd EMBL Science and Society Conference on One Health to try and integrate human, animal, and environmental health. And if you're interested, then please do sign up by the 12th. And I already mentioned, we're at COP26. We even wrote a white paper. If you're interested, please do check it out, Harnessing Molecular Biology to Accelerate the Green Recovery. And with that, I will thank you for your attention and take any questions that you might have.
Thank you, Edith. That was an interesting presentation. So the idea is that we would have all presentations realized and then have a discussion session at the end. So I understand that Kevin Tyler is around now. So he is the chair for this session and the chair for the Ecology and Environment Grand Challenge of the Euglena Network. So Kevin, I will pass this on to you. So
thank you,
carry on.
And apologies to people for connection issues. Just brought me in a couple of minutes late there. Okay. Okay, so fantastic. That was an amazing talk from Edith, and so we'll have some questions and answer with her later on. But if I can, if we can move to the next speaker, please. Are we there? Is it? Can you share your screen? Okay.
Yes. Good afternoon. So I suppose I'm the next speaker.
Sorry, Maja. Yeah, is it Maja? Maja.
Yes, it's just Maja.
I have a Tunisian student who spells her name the same way but pronounces it Maja.
Yeah, I heard it many times. So. Okay, so I'm sharing my screen, and then I'm starting the presentation. Okay, I hope it works. So. My name is Maja Łukomska-Kowalczyk. I'm from the University of Warsaw, from Poland. And I would like to thank the organizers for the opportunity to talk here. And I'm going to talk about environment and ecology of euglenids. And just before the beginning, almost all euglenids are freshwater organisms. We have only 3 genera, Euthretia, Eutretiella, and Rapaza, that occur exclusively in marine, on brackish water, but most of other species and other genera are freshwater. And despite, even though euglenids possess chloroplasts, we should remember that they are not fully autotrophic. In fact, they are photoauxotrophs. That means that they need some organic substrates. Okay, so they need some organic substrates for growth, and they can utilize some fatty acids, some sugars, and other simple organic compounds produced by bacteria. So they usually occur in some environments that are rich in decaying organic material. So they are these bacteria. So they usually, the typical habitats are in fact small, shallow bodies of water, especially that bodies of water that are contaminated by some animal pollution. So we usually can find euglenids in some farm ponds, like duck ponds, watering places for cattle, some field ponds, some aquaculture ponds, sorry, I don't have all of my screen, so okay. And we also can find them in other types of ponds, in public parks, and even in some hoof prints of cows. And some euglenids can occur in extreme conditions, like Euglena mutabilis, that occur in, for example, in peat bog ponds, so in very low pH, like about pH about 1. So, but in general, most of euglenids live in like very many kinds of small bodies of water, and recently these small bodies of water are very intensively investigated because, in fact, they are very abundant in Earth. It's like many millions of them, and they are very diverse, and they are considered as very important ecologically, as some biodiversity hotspot of many groups of eukaryotes. And the euglenids can be very important components of the microbial loop and in general of food webs in those small bodies of water. And they can form really dense populations that can even form blooms in those small bodies of water. So they are very important element of very important types of bodies of water. And it's why we would like to know more about them, about their distribution and so on. But now our knowledge is kind of limited. We don't know many about distribution of species. We don't know about some taxa preferences of some taxa, about the population dynamics and so on. And of course, we have some knowledge, but it's mainly based on rather old studies based on morphological identification of species. And in case of euglenids, it's somehow difficult and time consuming. So we don't have many information about this case. But of course, nowadays we have DNA metabarcoding that is really fast, and method of molecular identification of organisms. It's in general, it's kind of easy. We can do sample from environment, isolate DNA, amplify this barcode sequence, and based on reference database, we can check the species composition in our sample. But in case of euglenids, it's not so easy. They are usually underrepresented in environmental metabarcoding surveys that targets like all eukaryotes. It's because we know euglenids are very special, and they have a lot of features, special features. Well, one of them is they have really long and variable 18S DNA sequences, and some variable regions of 18S are used as eukaryotic barcoding sequences. And it's, for example, probably the most important is P4 hypervariable region of 18S DNA. That is a universal barcode for all eukaryotes. And in euglenids, this fragment is like, in average, is like 600 base pairs long. So it's a little bit too long for for sequencing it with Illumina. So it's one of the problems with euglenids. The second problem is that these variable 18S rDNA sequences don't match the universal primers very well. So we have problems with amplification with standard primers. And of course, we don't have a complete database of reference sequences of euglenids. And the third problem is the environment of the euglenids, these small bodies of water that are very rich in organic matters. This organic matter can clog the filters, it can contain PCR inhibitors, so we have a lot of problem with the metabarcoding of euglenids. So we decided to design euglenids targeted DNA metabarcoding approach. So just to solve all of these problems. So now with this approach, we know that in fact the filtration can be replaced by centrifugation. We know that the best solution for DNA isolation are soil dedicated kits. And maybe the most important thing is that we designed a euglenid specific DNA barcode. It's V2 hypervariable region of 18S rDNA. It's shorter than V4 region, so it's easier to handle. And of course, we designed the euglenid specific primers to amplify that region in euglenids. And we also changed we have also our database for identification of sequences. In fact, we use the SILPA database, but with the addition of curated sequences of euglenids. We've verified the taxonomy and so on, and we also added some species of some sequences of species that weren't in this database. So we tested our methods at first on one sample. And here you have the results of this experiment. For the beginning, this graph with green bars. These green bars are a fraction of reads that we obtained that belong to Euglenida. So in average, it was 85% of Euglenids reads, so quite a lot. And almost all of them were identified at species level. So was also okay, and we found like 50 species in this one body of water, and among them were 20 of 22 species that was observed using microscope. So we detected almost all of them, and we also detected 30 species more. So the method is working, so we decided to use it in bigger environmental survey. So we've chosen 20 bodies of water that are located in 3 regions of Poland. You can see it here on the map. Among them were some field ponds, farm ponds, fish ponds, and other kinds of ponds with euglenids. And we were sampling during 3 years, several times a year. So in total, we obtained 300 samples. We amplified our DNA barcode sequence, V2-18S DNA fragment, and then we sequenced it. And using this database with curated euglenids sequences, we just now know with the taxonomic composition of the samples and biodiversity of euglenids and so on. And I'm going to show you some of our results. So in the whole study, we found 163 species, and that is quite a lot because in our database there was about 200 of species, so we found really a lot of them. And in each sample we had like from 10 to 66 species, so it varied between bodies of water that we analyzed, but in general there was a lot of species in most of samples. And we found like representatives of all of freshwater in general, the 12 of them. but most of the reeds belong to the genus Trachelomonas. It was, well, maybe not most of them, but like 44% of them belong to Trachelomonas. You can see it on this graph. Here you have fraction of reeds in analyzed water bodies, and these brown bars are reeds of belonging to Trachelomonas, so it's really a lot of them. And the next genera was Euglena. We had like 13% of reeds of Euglena, then some other genera. And when we look at the number of species detected from the genera, it looks kind of similar. We have 38 species of Trachelomonas, and then many species of Lepocinclis, Phacus, and Euglena. And but now we are looking at species. So among these 163 species, we have 31 species with a fraction of reeds higher than 1%, so kind of abundant species. And here you have a list of those species with a fraction of the reeds. And you can see that the most of them are in fact species of Trachelomonas. So the most abundant species was Trachelomonas intermedia, species of Trachelomonas of a genus with lorica around its cells. So you can see some cells of Trachelomonas intermedia here with very dark lorica. We hardly can see the cell inside it. And we also have some very abundant species of Euglena, Euglenaria, and some other genera, but we don't have any abundant species of Cryptoglena, Eugleniformis, or Strombomonas. There we have only very rather not numerous species. And the next thing is I made a phylogenetic tree of all of the species that were found in the study. I will show you a little part of this tree. That is the part with species of Euglena that were found. And on this tree we have a lot of dots. Each dot is a sample. Is a sample with that species found. So if we have a lot of dots on this tree, it means that the species was found in many samples. And the color of the dot corresponds to the pond.
Are we just running well over at the moment?
Excuse me?
We're just well over time at the moment, so.
Okay, so then maybe I show you the most important thing, because we have some common species with a lot of dots. They were found in a lot of samples, and but I showed them to you earlier. But we have also very rare species, and among these rare species, you can see some very well-known species like Euglena viridis, Euglena hiemalis, and Euglena gracilis, that in fact was found only in 3 samples out of 172 that was in this analysis. So it was kind of surprising maybe that our model organism is in fact very rare in environment. And I have also, we also would like to know what factors determine the taxonomic composition of our samples. So here you can see multidimensional scaling graphs with sample colored by a pond they are originated from. And in fact, we can see that samples from the same ponds group together, and we didn't find any grouping by month or year or region and any other parameters. So in fact, we have rather stable populations of Euglenids, stable composition of Euglenids during the whole period of our study. And as a summary, maybe we have this metabarcoding approach that works. We found a lot of Trachelomonas. We found hardly known Euglena gracilis in environment. And in fact, we found a lot of species in Poland that is not a very big country. And I would like to thank all my colleagues from the Institute of Evolutionary Biology from the University of Warsaw. And I would like especially thank Professor Bożena Zakryś for this photos of euglenids I had on my presentation. And thank you for your attention.
Thank you so much. Fascinating presentation. Again, we'll take questions when we're through the final presentation for today.
Okay, so let's stop sharing
this session. So I think that's Peter. Did you want to? Can you share your screen? Okay.
Sure. Yeah. Can you hear me?
Yeah.
Hello, everybody. All right. Let's get started. I will share my screen. Thank you to the organizers for inviting me. It's really great to be here. I just realized that we're going in this session from the biggest biotech organization in Europe to the smallest, I guess, because I'm doing independent research on Euglena on my own. So it's quite a big contrast to the EMBL. So I look forward to the discussion. But I will take you in this presentation through some of my work on gaming applications with Euglena. This is a side project of mine, which I've been working on for several years. First, I worked at an institute in the center of Amsterdam. It's called WAAG. It's an independent organization. And there I started a laboratory, a citizen science laboratory, which was called the Open Wet Lab. It's a place where people could come over and do their own projects. And we even had a GMO license to do genetic engineering and did all kinds of projects. We had open evenings every Tuesday where people could come by and do biotech experiments ranging from neuroscience to working with pigments and bacteria in fashion, all kinds of projects. And at some point, we opened even a store in the center of Amsterdam and where we sold microbes, all kinds of different species for just people to use at home. And of course, some were very popular, like spirulina and myceliums, but also Euglena was one of the favorites in the store that people would like to use and play with. And also at WAAG, I started the Biohack Academy, which is a program in which I taught people how to build their own labs using open source hardware, anything from incubators to PCR machines and spectrometers and photo bioreactors. These are all freely available. The designs are freely available online. And I've been teaching this course all around the world, from Brazil to Japan, even in Iran. Taught people how to build their own biolabs and do these type of experiments on their own. And one of the things that was included in this course was biotic gaming. And well, some of you would already recognize the Euglena on the screen. We will build our own hardware and interact with these organisms. And of course, before going into the details, I of course have to say a big thanks to Ingmar Riedekuse, probably is well known among your community, with whom we collaborated back in 2016 on creating a workshop to teach people how to build these type of machines themselves. You probably know his work, where he showed how you can interact with these organisms in game settings, and we created smaller open source kits that everybody could build themselves and do these experiments in public. Nowadays, I do these things at home. I have my own little small fab lab. This is a laser cutter and a 3D printer, which is in the room next door. And I prototype microscopes. This was an early prototype. This is another one where we created an entire instruction kit on how to build one yourself. And well, there have been many improved versions. And I wanted to show you the latest developments in this area, but maybe it's nice to use my Instagram for that. Currently I have made a device in SketchUp. So this is a free program where you can 3D design your own hardware. And in this device, you can control LEDs with a PlayStation controller. And well, you can play games like you can see in this video, where you are a spaceship in between the Euglena, and well, one player is controlling the lights, so controlling the movement of the Euglena, which are of course phototactic, and the other player is controlling the virtual player on the screen. And this game is on my website. It's called wetwareworks.com, and you can go to this early release. I don't know if it works while I'm also video presenting, but it works in the browser. And let's see, does it work? Oh, yeah, it works. Well, basically, you are the small boat over there, and then when it hits the microbe, the game is over. This is also online, freely available. You can edit the source code if you want to change anything in that game. It's on GitHub, and organize workshops. I provide kits for building these type of devices. And on my website, I also have a long read on all kinds of biotic games that other people have made to illustrate that there's a long tradition in creating video games with microorganisms. And this was actually one of the early versions in Oslo that I made, and I noticed that people find that it's a lot of fun to interact with microbes in this kind of way. So yeah, I think that's what I wanted to share with you. I think this is a nice way for people to show, to learn about microbiology in a playful manner, to interact with these organisms they probably would never encounter otherwise, and also to start conversations about the role of microbes in environmental issues, but also about science, how you can also study these organisms using microscopy and interactions. So yeah, I hope you enjoyed my presentation, and look forward to any questions. Thank you very much.
Thank you, Peter. That was very entertaining. Okay, so I've got a few questions coming in. I'll go back to Edith initially to ask one that's come in from Julius Lukes, who's here. So I think probably a lot of people actually speaking for here, he's really looking at how does our community specifically access some of the resource that you have at EMBL. I think you outlaid a fantastic vision of on a grand scale, really. So we're a very small community with people who'd like to participate in that vision, but how do we do it? And is there direct access to some funding, or is it sort of through facilities?
Yeah, I wish we could just provide funding too. We're actually looking for our own funding right now. To talk to you about it, I have 2 weeks, and it's been like a marathon for the last 2 years. Trying to get 27 countries is like herding not cats but hyenas. Anyway, so we hope that we will have enough funding to do this program, and the way that you can make the most of it is by reaching out to us. Planetary biology, for example, or microbial ecosystems, they each have chairs or co-chairs, and there are a number of initiatives that have already been set up. That goes from, as I said earlier, there are these sort of sabbatical programs. We have postdoctoral programs, so shared postdocs, and we really want that to be sort of completely fed into this new program. We have openings for group leaders if people want to actually come and work with us, and we will provide access to these new facilities. So that is, I think, the key to this. For a small community working on a sort of not common garden organism, this is exactly where we could hope we can help, because we can provide, for example, the technologies to measure single cell metabolomics and combine that with imaging and combine that with detection of environmental factors, chemical, physical. That's exactly what we want to do. And one of the co-chairs of planetary biology is actually the person in charge of our core facilities, Raina Peppercock. So for that area, we have a program manager. I can put her email into the chat, And you're very welcome to reach out to her, because right now, as you probably know, there are a number of calls for example, Horizon Europe funding, where people are joining forces, and EMBL is very good at acting as sort of glue. So we've already just deposited a big program on it's called Bio Ocean 5D or something like that. And there could be other opportunities, or it could just be, one-to-one projects. And we're very happy to work in a very sort of hands-on way, very collaborative. It's always fun to work with people at EMBL, you'll see. And we always find ways of finding money. So even if we don't have much ourselves, we're very good at helping leverage funding as well. So please reach out to us, and I'll put the name of Giuseppe, the email of Giuseppe into the chat.
That would be great, because I know there are other large organizations that people will have worked with, people like Tres Cantos, which has a very nice model for doing some of this kind of dissemination of expertise into communities. And so I think a lot of what they do is through named people, named advocates. So if we can, if you can give us some names and some websites and things, that would be
absolutely. And I should say that because the program hasn't officially started yet, we're already gearing up to it, but our funding will only start in January. We haven't been that vocal on our website and everything, because we didn't want to sort of jump ahead of ourselves, but it's definitely going to happen, however much funding we get. So watch our website as well, is what I'm saying. Even if the links are not there yet, they'll be coming out soon. But I'll put Giuseppe's email in the chat.
Okay, so that's great. If I move now to Maja, I've got one here from Vladimir Hampel, who says, were you able to establish a clear species boundary measured in V2% divergence between traditional morphospecies, or is it a continuum? And if yes, was the boundary similar to between genera of Euglenoids? That's a very specific set of questions, but I think maybe you could say a little bit more in general about how you recognize species as well, perhaps to provide a bit of context.
In fact, in this study, I didn't establish boundaries like with a percent of divergence. I just classified my breeds to species based on this reference database and using the naive Bayesian classifier. In fact, it was a classifier in China. So I didn't check this percentage divergence here, but in fact I did it earlier, and I had clear species boundaries in some groups of species, and other there wasn't. It wasn't impossible to have it boundaries, but yeah, this time I didn't do it this way. It was like the identification of species, and then based on identification, I know if it was the same species or various species.
Okay, very good. Okay, so I've got one here for Peter as well. Neil Emery says, who is your typical clientele? Teachers, academics, amateur biologists, or others? I can see educational value and just plain fun. So he's wondering who you're interacting with primarily.
Yeah, that's a good question. I've been connected well to the whole maker community around the world. So there are many maker spaces and fab labs, fabrication laboratories in hundreds of cities around the world, where people create their own hardware, electronics, and they often find that the combination of biology and creating your own hardware is an interesting one, and this is my typical, let's say, group of people that I interact with. But also, of course, I've been at schools demonstrating and also giving workshops, museums, also about science museums, and also design museums, or more like interdisciplinary places that feature bioarts or biodesign. So it's quite, and also in the gaming scene of actually there's also an interest in this type of development. So it's actually quite broad, because it combines so many different fields. a bit hardware development, electronics, programming, biology. So it's a really combination of all, let's say, traditional fields of science. So yeah, it's could come from anywhere.
So you have great outreach into the general community as a result of doing this sort of thing. And you talked a little bit about doing some level of citizen science, at least in terms of getting people to construct this hardware from your instructions. Have you thought about whether you can enable any sort of wider scale citizen science projects with what you've done so far? Do you have a real world, perhaps, application which could create some impact?
Yes, sure. Yeah, when I worked at WAG, we often were part of European Union Horizon projects on citizen science. So some of the projects we did really scale to European level. Mostly those were, for example, about environmental sensing in the city, air pollution and noise, but also biological ones. We once did a big project on trying to identify antibiotics by doing some simple screening experiments, and that was really picked up well. I think we went all around Europe at least.
So where people are thinking about using Euglena as a biosensor, for instance, then and can sample for you, then you would have a mechanism there essentially to provide a readout from anywhere that people are set up to do it.
Yeah, that would be a nice idea. I'd be open to continue the conversation on that, but also by combining it with gaming, it also draws people in that maybe would otherwise not be attracted to these type of activities. So yeah, combining science and scientific measurements with fun is, I think, a very powerful way of doing things.
Edith, you've got your hand up.
I just wanted to say something to that, Peter. That sounds like a fantastic idea, and in fact, I forgot to mention it, but within the exploratory part of what this TREC program is going to be in our new program, we want to do citizen science. Actually, Tara is already doing citizen science, giving kits to people, trying to make sure that, for example, just your standard fishing boat, when it goes out, it can actually have a sampling kit. So now this is for collecting samples at certain depths. But it sounds like this is something maybe we could join forces on if you'd be interested. So maybe we could discuss starting January if you'd be interested to. Sounds good.
So then let's connect. Yeah.
I like that's happening in real time here. So I mean, I think also that because both of you have these great networks outwards in terms of outreach, that a lot of the laboratory-based kind of contingent are quite interested in how best to do that. And it sounds like these are both good people for people to connect with if you want to show how you're getting message out and then to collect data back in through that route. Okay, so I haven't got a lot more questions in the inbox. If anybody has any questions that they'd like to raise themselves with their voices, switch your microphone on now and feel free to do that. Pika's go ahead.
Yeah, thanks. I would just like to say these were really excellent talks and interesting how these things link to so widely. I mean, from the scale of where we are looking at, but also this gaming and how to do the outreach. So I would just congratulate everybody, and this is we should just all of us jump into all these activities in this way that they go forward. I had one practical question to Edith, because you're widening your scope. So are there some specific groups that are difficult to reach, or who you would like to have more actively involved because it's so large? Is there some depth?
I mean, I think what we've realized is that there are some communities of scientists who don't necessarily have the kind of biology that we do in their radar. So, for example, some of the approaches that people use, the genomics, metagenomics, of course, that's all there and people are using it. But we're trying to say, hey, we can actually provide even better technologies. We don't want to do your experiments for you, but we want to try and help you do experiments in the most modern and effective way possible. So it's about reaching out to these communities of scientists so that they can, building up trust and also showing them that we're useful. So far, I have to say, we haven't had pushback from anyone. It's just about building those bridges. A lot of people warned me at the beginning, this started about 3 years ago now, watch out, there'll be communities who don't want you to step on their toes. Actually, not at all, because we're an organization and because we're there to serve, that's what we were invented for. People are actually usually very welcoming. It's just a question of making sure they understand what we're aiming for. So this was a fantastic opportunity for me to tell you about what we're aiming for and invite you to join.
Yeah, I think, sorry, Kevin, should I just add one more thing to the
Yes,
biosensor?
please do, thank you. Yeah.
I think the point you raised about the biosensor is one area I actually see Pieter's work actually connecting to this. I mean, it's really be nice to see how this works on scale, because for years there has been some publications and efforts in that regard. But I'm not sure that I've actually read or seen any active development. I mean, this is one area that it might actually work to try to connect the engineering and the biology. And I'm sure, apart from the work Pieter just talked about, on Thursday, we'll be hearing from Igna as well on some of the additional things that are being done. So maybe, Pieter, this is something within the community, again, if you link up with Edith to see how this could potentially work, really. So.
There is one more comment from Ainsley Lewis, which is really, okay. He's withdrawn his comment. He was asking about genomics, but it was going to bring me really to some of the points that Maja raised, which was about diversity of Euglena and the environmental role of that diversity. So she's managed now really to give us a good feel in the areas where she's been looking of just how diverse that community must be. Is probably a kind of a minimum value. I don't know if she would agree with that, but we begin to get some genetic information, but very little phenotypic information. And I wondered how people felt what was the most appropriate way to start to gather some information about the roles of these many different euglenoids in the environment, and should it be a predominantly sort of genomic, metagenomic approach in the first instance, or do we need to get down into the biochemistry and the cell biology of these organisms in order to do that? Maybe Mike can comment on that to lead us off.
I'm not really sure if I'm the right person to comment it.
Do you have plans to explore beyond the genetics here and the diversity that you reveal and to figure out some sort of functional and phenotype on these organisms? Or are you mostly just interested in surveying diversity?
Yeah, in fact, I'm rather interested in the biodiversity of euglenoids, about the factors that influence it, and yeah, I think I'm. It's I think my part. I just yeah. I think that's that's it.
So I think we all think that Euglena, part of the reason we're all here, right, is that Euglena is what Edith would have referred to as one of her key organisms, right, that it has a key role in the ecosystem, but we are all looking at it through a prism of Euglena grassalis. And I know Julius and others are working hard to bring us a little bit more information about a handful of more species. But possibly, I think the implication of what Maja showed us today is that we need to cast our net wider in order to appreciate what kind of environmental effect this organism is having, or this group of organisms A is having, and B is capable of having, if we are thinking about how to exploit it best. Is anybody working along those lines? Does have any comments along those lines? I'll put it out there. So Edith has written, combining imaging and metabolomics are approaches that would be able to apply in combination and go well beyond genomics.
Yeah, I actually skipped that slide because I was already well over, but we've done some pilot projects, not on Euglena, but sampling in very precise levels in the ocean, because we know what kinds of plankton we're after. And there, the whole approach is to not just sequence, which is what was done for Tara Oceans, but to actually do imaging, to collect samples and visualize them and measure where possible, for example, metabolic activity. And even we did this project called Freeze on the Beach, which actually consisted of taking the equipment to do cryo-EM on diverse samples that we were collecting. And that really gave some incredible insights where you see supposedly similar organisms with very, very different structures, behaviors, internally, the number of chloroplasts that were there. So I really think that we now have those technologies. And the wonderful thing about all of you is that you're all interested in, Euglena. Going out and looking at all plankton is rather vast, but, some people have special bits of the sea they're interested in, but this is typically the sort of thing where I think an organization like EMBL could help. So as I put in the chat, reach out to Rainer Peppercott. He would love, I'm sure, to dream with you and come up with approaches to combine different types of technologies, so you get more information out of your diverse populations.
Well, we had a couple of hands up. We have one dreamer, and Scott Farrow, who has put his hand down now, I think, or at least he's disappeared from my screen. There he is. Do you want to go first, and then we'll have Barry after you?
Sure. I think the EMBL is a fantastic resource to help us pursue some of these grand challenges. I'm curious, first of all, is there any connection with international research agencies currently, so countries like Canada could get involved and take advantage of your facilities?
Absolutely. I mean, we don't, we're kind of open to all. Our member state funding allows us to provide resources that all of you use probably without even realizing. EMBL EBI, obviously everyone uses those data resources, and that's 27 European countries who are paying for that. But we also have lots of initiatives that are across continents. And indeed, if there's a special interest with Canada, we could definitely link up. For example, I was just in touch with the Biosphere 2 people in Arizona who are very keen to team up with us. And we signed an MOU. And here, they're kind of looking for projects. And so we have people who are looking for ecotrons, or what they call mesocosms. So if you know that there's something that's specifically of interest, be it through training or use of facilities or building up some kind of new technology, then reach out to EMBL. We're very open to other continents for sure.
Barry, did you have a question?
I was just wondering, and Maja, if when you detected these species, if you actually looked to see if they had specific partners when you were looking at your euglenoids, right? Because we know that there's a lot of metabolous strains that we've been working on that have partners you can't get rid of because the strains don't do well. And so it would be a very fine scale version of what Edith was talking about. It would be a very localized Euglena plus a couple of partners, which wouldn't be a mesocosm yet, but it would be the start. So did you find any of those, Maja, when you looked at the euglenids in situ?
So in this study, no, because I used like euglenid primers, so I can't like have any other sequences. But when we try to have cultures of euglenids, they always have some bacteria in it, but I'm not sure if it's any specific bacteria or just bacteria from water. So in fact, I'm not sure if I know anything about their partners now.
Mutabolus has a fungal partner as well as bacterial partners. So, but you're right. Specificity would be the kind of thing that would be kind of interesting to look at. But
yeah, I think it would be interesting.
I think that's where a great project could emerge with the EMBL using microscopy in real time, combined with sequencing and omics platforms to really dig a little deeper there. I think that's an opportunity.
And Barry, have you looked at viruses in euglenoids?
no, I haven't.
Because there must be some, and they must be having an effect.
No, there's not. No, viruses are in every form of life except Euglena, right? Yeah, that's interesting.
I feel I need to jump in here.
We should be able to do it.
Project bigger, Barry.
No, we're going to set up a team, Emma. Don't worry.
Okay. Okay.
God's going to fund it.
Works for me.
Okay, so let me just draw people's attention. Edith has put in a reference of an example of a collaboration with the EMBL into the chat, so people should have a look at that. I think unless there's any other burning questions, we're coming up against our time for this session. So let me thank our amazing speakers again. There's a fantastic and probably the most diverse set of talks I've ever hosted. So that was great. And hopefully we'll see you for the next session shortly. Thank God. Did you want to come in and say anything?
Yeah, I would like to thank Edith, Maja, and Pieter for actually agreeing to come speak to us, because one of the things in vision here is for us to begin to see how diverse the application of Euglena can be in terms of the biology and application and so many other aspects. And I think some of this actually touch beyond this. So this is the end of the session. I think Kevin, if that's okay, then we'll have the second session starting in 20 minutes time, or that should be at 16:30 my time. That is in the next 15 minutes. So I will pass this on back to Kevin, and thank you again, all the speakers, for actually coming. We'll continue this conversation and hopefully follow up on some of these points, basically.
Okay, great. So I'll see you all in 20 minutes. You have your comfort breaks, and when you come back, Ross Lowe will be moderating, I think, isn't that right?
Yeah. No, I think we're back in 15 minutes. So we come back at 16:30, which is in the next 15 minutes. Okay. So when we're back, then Emma Kaszecki will moderate the second session, basically.
Okay, perfect. All right, thanks very much.
Thank you, everyone. Feel free to just hang around or just, talk to people. Thank you.
Okay, bye-bye.
Hi, Emma.
Hi, thank God.
Hi, Barry.
Hello, Emma. Hello, Barry.
Hello,
Oh,
hi.
thank God.
Hey, hi, how you doing?
I'm good. How are you doing, Barry?
Not bad, not bad. This is going well, thank God. This is really done well. Thank you, thank God.
You're welcome. So I think we can just move quickly with the next session. So thanks to everyone who has attended the last session. That was really engaging. I mean, lots of things to actually follow up on for those already working on them and would like to follow up. And this connects with the vision of the network on bringing the community together, discussing all of this through the annual Congress and looking at the next steps and on how we advance euglena science. It's been a fantastic session, Ecology and Environment, and thanks to Kevin for chairing the first session. So over the next 1 hour, we're going to be listening to speakers, oral speakers, talking about their work particularly, and that will be chaired by Emma Kaszecki. Over to you, Emma, please.
Thank you, thank God. So our first speaker is Jie He, talking about metabolic responses in Euglena gracilis. So whenever you're ready to share your screen.
I think Jay, actually, I'm not sure if he's here, but he did send
the email.
An email that an apologies that he will not be able to make it today. So we can just move to the next presenter, please.
Okay, this is Saleha Khan. Whenever you are ready to share your screen, talking about aquaculture and euglenoids in aquaculture.
So do we have Saleha Khan? Is Saleha Khan on the call today?
Yes, hello, good evening.
All right, go ahead, go for it, please. Please share your screen if you have a slide to present to us.
Yeah, so this is okay. It's okay. Can you hear me?
We can hear you.
My slide?
No, we can't see your slide yet. So at the bottom of the screen, you will see share screen. Then tap on it. It's just in the middle, at the bottom in the middle. When you tap on it, it will bring up a window at the top left. Then you click desktop 1. Then you can start sharing your screen.
Slide share.
Yes, yes.
Okay, thank you so much.
Perfect. All right. So maybe you want to share the first slide and then we can move on sequentially. All right, perfect. Thank you.
You are welcome. Just a moment, please. Okay. Honorable Chair of the session, respected organizers, moderator and reporters, and respected scientists, good evening. I'm going to present my research work on ecology of harmful euglenoids in aquaculture ponds of Bangladesh. Welcome to my presentation. What's the problem? Okay. Welcome to my presentation. Though euglenoids have various powerful benefits ranging from health, cosmetics to sustainability, but today I'm talking about its harmful side. That means negative impact on water quality and aquaculture. So before talking about my research findings, I would like to say about the background of my research work briefly. We know that the impact of aquatic environmental pollution is being seen through a variety of disturbing symptoms, including an worldwide decline and collapse of fisheries. According to the World Health Organization, while a combination of factors, most notably overfishing, are to blame, algal pollution is undoubtedly a significant contributor. We know that though the microscopic planktonic algae are critical food for commercially important crustaceans and fishes, however, in some situations, algal blooms can have a negative effect, causing severe economic losses to aquaculture and fisheries and having major environmental and human health impacts. Mortality of fishes, irritative bad odor from decayed algae and bad odor from fish muscle, even after cooking during harmful algal blooms have been reported from different countries. Algal pollution is producing increasingly in Bangladesh. Pond aquaculture plays an important role in the livelihood of the rural poor. It is therefore important that the aquatic environment should be used in a sustainable way and that the resource base shouldn't be damaged or destroyed. But unfortunately, water quality deterioration, especially euglenoids and blue-green algal blooms in aquaculture ponds in Bangladesh, is becoming more common and producing increasingly adverse impact on aquaculture. Now I have come into the main point, and this is harmful euglenoids and their impact on aquaculture in Bangladesh. Here in our country, the main problem is in order to get higher fish production within a short time, fish farmers follow intensive and semi-intensive aquaculture, utilizing high doses of fertilizers and fish in confined waters year after year. And as a result, most of the fish ponds have become hyper-nutrified. Moreover, some people of the country dump waste from recently boosted poultry farms in open places. During the monsoon months, the surface runoff water carries organic matter from different sources to the open water system. We know that euglenoids are tiny microscopic organisms. Though there are about 40 genera and 800 species of euglenoids in the world, in Bangladesh, we have identified a good number of species of this group of noxious phytoplankton, of which these three genera like Euglena, Phacus, and Trachylomonas are mostly abundant, and some species of Euglena are making serious problem in our freshwater ponds and lakes. Euglenoids are considered as plants due to the presence of their chloroplast and their photosynthetic activities. On the other hand, they are also considered as animal due to the presence of their flagella and their locomotory activities. Though we are facing serious problems with the blooms as well as mortality of fishes, due to this notorious, noxious, harmful and toxic algae, there was no comprehensive study on the morphology, ecology, toxicology, and management of this group of phytoplankton. Therefore, the occurrence, abundance, morphology, and autocology of euglenoids are needed to be thoroughly studied. Our main objectives are to investigate the ecology of euglenoids, and the second one is to study the impact of duckweed and periodic liming in water quality maintaining and in controlling euglenoids bloom. This is our experimental design. The experiment was conducted in 12 experimental ponds under 4 treatments, namely T1, T2, T3, and T4, with 3 replications in each treatment. T1 and T2 are assigned to the ponds treated with duckweed and lime respectively. T3 was assigned to the ponds treated with both duckweed and lime, and T4 as control ponds. Materials and methods. Due to lack of time, I'm not going to explain about materials and methods. Here only I'm telling that the methods used for analyzing different water quality parameters, then identification and quantification of euglenoids. Now let's have the key findings. In the present study, 3 genera of euglenoids, like different species of Euglena, Phacus, and Trachylomonas are recorded in both experimental sites. That means in our university experimental ponds, as well as in fish farmers' pond. Morphological change in the present study we found that euglenoids can grow everywhere, especially in fresh waters, but their abundance vary from place to place depending on the surrounding environmental condition. If the condition is suitable for them, most of the cells are spindle, but in unfavorable conditions, they have the capacity of forming cells. So in unfavorable conditions, first they start to be oval from spindle and then from oval to complete spherical. After forming cells, they just wait for the suitable time, and with getting favorable situation, they start growing vigorously again. Different environmental parameters have a strong influence on the morphology of euglenoids. Morphology of euglenoids is variable and strongly influenced by pH. At acidic pH, euglenoids are found to be mostly spindle-shaped. And with the increasing of pH, spindle-shaped cells of euglena started to be oval. At alkaline pH, cells are found to be changed their shape first from spindle to oval and finally from oval to spherical. Color. Euglenoids have another characteristics. In fact, euglenoids have a peculiar characteristics. They have red pigment in their body. During strong sunlight, they just can bring their red pigment from their inside of the body to surface of the body to protect themselves from ultraviolet rays of light. When the light falls, then the red pigment enter into the body and the chloroplast onto the body surface for photosynthesis and food production. That's why the color of euglenoids bloom looks brownish in the morning and become bright red during strong sunlight, and again pale reddish before sunset. This is very much clear from this slide. Influence of nutrients or the morphology of euglena. At high concentration of nitrate, nitrogen, and phosphate, phosphorus, most of the cells are found to be spindle. And with the decreasing of nutrient concentration, they started to change their shape. For your kind information, harmful algal blooms has been endemic in Bangladesh and has become widespread in recent years, which is seriously alarming to the aquaculture. If we look at your traditional or commercial fish pump ponds, then only by observing the color, we can easily understand that the water of most of the ponds are polluted by euglenoids or blue-green algal blooms. So there are two types of species. Some species can kill fish. There are two types of harmful algal species. So we know that some species can kill fishes by oxygen depletion, and some species can kill fishes by potent toxins. Now list the impacts of darkweed and lime on the abundance of euglenoids. In this figure, we can see the cell density of this phytoplankton group was comparatively higher in the control ponds. That means why darkweed and lime are not applied throughout the study period. In T1, T2, and T3, the cell density was comparatively lower in T3. That means where lime and darkweed treated ponds in mid-June. Here in this figure, we can see the abundance of euglenoids in relation to pH. The abundance of euglenoids in relation to different pH values showed that the cell density increased at acidic pH and showed a declining trend with increasing pH value. The highest cell density was recorded in the ponds of T4, that means in control ponds, when pH was around 6.60. And now, here, in this figure, we can see the relationship between euglenoid cell density and nitrate nitrogen concentration. In experimental ponds, during the early bloom of euglenoids, nitrate nitrogen was high and there was a decreasing tendency of nutrient concentration from peak bloom period. Here, in the next slide, here we are showing only bow experimental ponds due to lack of time. But in both bow experimental ponds and farmers' ponds, during the early bloom of euglenoids, phosphate, phosphorus were high and there was a decreasing tendency of nutrient concentration from peak bloom period. Now let's have the conclusion. We know that Euglenoids bloom can inhibit the light penetration, which hamper photosynthesis of other plankton, which causing lower dissolved oxygen. And as the Euglenoids grow abundantly and form bloom in nutrient-rich water having acidic pH, so the combined effect of fish edible floating macrophyte lamina, periodic application of lime, and stocking of some planktivorous species can control the excessive growth of Euglenoids. What we're faced with today is that the effect on public health and economic impacts of harmful algal blooms are now showing signs of a truly global epidemic, and we should start to respond to this problem as such. So, as aquaculture is threatened by algal blooms and the situation is getting worse and worse, we must find out a way how to utilize these alarming things as resources. If we can utilize it properly, it can be a good resource. Most importantly, people responsible for management decision on pollutant loading of waters should be made aware that one probable outcome of increased nutrient loading will be an increase in harmful algal blooms. Finally, I would like to say that we should try our best to keep our aquatic environment safe from any hazards, the final goal of which is to increase the production of fishes, manage and conserve fishes resources to sustain benefits for the present and future generation, to increase overall economic growth and generate employment and incomes, particularly for the rural poor and unemployed youth of the world. Thank you very much for your patient sharing. Thank you very much. Oh, finally, I'd like to say only some sentences, and these are, I'd like to say, as Euglena sanguinea and Tracholomonas sp are very common in our country, and we have already isolated these two species, and Euglena sanguinea can produce euglenophysine and can kill wild and farm fish populations. We're extending our research on the toxicology of euglenophytes and their impact on fisheries and human health. That's all. Thank you. Thank you very much.
Thank you for that talk, Saleha. So now that we know about the ecology of these aquacultures, we're going to hear from the next speaker about how we can manage euglenoids in these cultures. So Sunzida, do you want to share your screen?
Yes.
Saleha, you just have to stop sharing your screen.
Yes, yes.
So up at the top, there'll be a bar and it'll say stop. There we go.
Assalamu alaikum and good evening to all. I am Sunzida Sultana from Bangladesh. I am a master student under the supervision of Dr. Saleha Khan in Department of Fisheries Management of Bangladesh Agricultural University. At first, I want to thank all the event members of first annual International Congress on Euglenoids for arranging this program. This is a good opportunity for us to know about euglenoids in the age of symbiogenesis. Today, my presentation topic is management of euglenophytes in aquaculture ponds for improving fish production in Bangladesh. Bangladesh is one of the world's leading fish producing country and ranked 5th in aquaculture production, but unfortunately, water quality deterioration in water bodies in Bangladesh is becoming more common and producing increasingly adverse impact on aquaculture. Over the last 2 decades, Bangladesh has experienced aquatic pollution in aquaculture ponds from harmful algal bloom, especially by Cyanophyta and Euglenophyta. That's responsible for declining fish production and human health. The most common bloom-forming euglenophores in Bangladesh are Euglena, Ficus, and Tracholomonas. Here we see some pictures of euglenophores bloom and fish mortality in different water bodies in Bangladesh. Reddish bloom of euglenophores is very common in fish ponds of Bangladesh. During heavy bloom of euglenophores, the fish breathe with difficulty at the surface and die because of oxygen depletion. Objectives of this research. The first objective is to investigate the impact of duckweed and lime in controlling euglenophores bloom. And the second objective is to study water quality and growth of fish in fish ponds with euglenophores bloom. Experimental layout. The experiment was conducted in 12 ponds under 4 treatment, namely T1, T2, T3, and T4. T1 and T2 are treated with duckweed and lime respectively. T3 was treated with duckweed and lime. And T4 as a control pond, where no duckweed and lime are used. Pond preparation. The experimental pond were dried, renovated, and exposed to sunlight for several days. The pond bottom are treated with lime at the rate of 1 kg per decimal. Urea, TSP, and cow dung were applied after 7 days of liming for fertilization, fish stocking, and feeding. Fishes comprising rui, katla, mrigal, silver cup, and Thai shortputty were stocked at the rate of 40 fishes per decimal. After stocking of fish, mustard oil cake and rice bran were applied as supplementary feed at the rate of 4% of the body weight of fishes per day. Sampling of fish. In ponds of all treatment, about 10% of stocked fish are sampled fortnightly. The length and weight are measured to see the growth. At the end of the experiment, all fishes are harvested to calculate their final growth and total production. Fish gut content analysis. For investigation of gut content, fish samples are collected at each month with the help of ShareNet. From each pond, 2 fishes of each species immediately after capture are preserved in plastic container with about 10% buffer formalin. From this table, we see that during the gut content analysis, 5 species of fishes in experimental ponds show they ate a variety of food items. 3 genera of euglenophores, 6 genera of cyanophores, 5 genera of basilariophores, and 12 genera of chlorophores were recorded during the gut content of fishes. The abundance of euglenophores in food treatments are shown in figure 1. The cell density of euglenophores was comparatively higher in T4 throughout the study period and showed a marked increasing trend from 15 July with highest value on 15 August. In T1, T2, and T3, the cell density was comparatively lower with a minimum in T3 in mid-June. The cell density of euglenophores in the ponds of T4 showed significant difference from the ponds of T1, T2, and T3. In figure 2, the growth of rui, katla, and mrigal in terms of weight gain showed variation in monthly observation and continued till harvesting. The mean weight gain for all species was found to be higher in the ponds of T3 and in the pond of T1 followed by T2. The lowest weight gain showed in the ponds of T4. In figure 3, silver cup and Thai shortputty was also shown highest production in the T3 ponds, around 2817 kg per hectare, and lowest production was observed in the ponds of T4, which was around 1726 kg per hectare. In figure 4, we saw that 4 groups of phytoplankton such as Euglenophyta, Cyanophyta, Chlorophyta, and Basilariophita were identified from the gut content of fishes during this study period. Comparatively less quantity of euglenophores are found in the gut of rui, katla, and mrigal. On the other hand, high amount of euglenophores was found in the gut of Thai shortputty and silver cup. with the highest value in T4 ponds. That was respectively 42.69% and 32.15%. Research outcome. From this research, we found that the specific growth rate of Katla, Rui, and Miguel were significantly lower in Euglenophores bloom-forming pond than the non-bloom-forming ponds. The growth of silver cup and Thai shortfooty were not inhibited by Euglenophores bloom. Mortality of fishes occurred in bloom-forming ponds where the phytoplankton community was mostly dominated by Euglena. Dissolved oxygen was observed to be very low during the period of fish kill, which was possibly due to the bacterial decomposition of certain dead Euglenophores. Application of lime and maintenance of duckweed will prevent Euglenophores bloom formation and provide better fish production and water quality. The grazing of Euglenophores by the silver cup and Thai shortfooty has some contribution in controlling bloom. Conclusion and recommendation. The bloom of Euglenophores in fish ponds of Bangladesh ultimately reduce fish production. The negative impacts of Euglenophores bloom can minimize by using duckweed and lime. Duckweed is used as fish feed and work as biofilter in pond water. It also reduces nutrient concentration from water bodies. On the other hand, lime keeps the environment alkaline, which is unfavorable for the heavy growth of Euglenophores. Both duckweed and lime are very cost-effective for Bangladesh. Also, a culture of phytoplankton feeder fish such as silver cup and Thai shortfooty prefer Euglenophores as their food item and biologically control Euglenophores bloom. So it can be concluded that the combined use of duckweed and lime is effective in water quality maintenance and controlling Euglenophores with good fish production. Thank you for hearing me and for your attention. Thank you.
All right, so we'll just have you stop sharing your screen, Sunzida, and we're going to move on to our next speaker, Hai, who will be speaking about metabolites in Euglena gracilis as a response to heavy metals. So Hai, whenever you're ready.
Hello, Emma. Could you hear me? Could you hear me well?
I can hear you, Hai.
Okay, thank you. Now I'm sharing my screen. Now could you see my screen now, right? Yeah. Yep. Hello, everyone. Today, I'm very happy to see all of you guys to be here to share our interesting study with Euglena. So my name is Hai. So now I am a PhD student at Dartmouth College, and I just finished my master study at Trent University. And in this project, I also work with Euglena gracilis, and I'm a student from every lab at Trent University. During a master study, I work with Euglena gracilis. You can see the picture of my organism in slide. And the project as well as identify the potential function of phytohormone in Euglena with 3 kinds of heavy metal: nickel, lead, and cadmium, and to see like whether Euglena tolerance with this metal or not, and how phytohormone improve Euglena tolerance to the heavy metal, and the possible application of phytohormone for heavy metal stress response in algae. Yeah, so I come up with some background of why we come up with an idea to develop this project. Euglena gracilis, for a long time, by more than 30 years, we know that Euglena gracilis have a wide ability to tolerate with a wide range of heavy metal, such as zinc, cadmium, nickel, and lead. And during this time, researchers also found that Euglena not only can tolerate with a wide range of heavy metal, but they also be able to develop mechanism during evolution, how to tolerate with metal by biosynthesis of phytokeratin, like metal binding compound, or like with sorry, I see message that you cannot hear me, or like. Oh.
Yeah, so I think there is a background noise coming from maybe I'm not sure if it's from your end, but yeah. So.
Okay, so how about now? Sorry, let me stop sharing. So how about now? Could you hear me clearly?
You just go ahead, basically, I think, so that we don't spill into others' time. So
okay.
It's fine now. Yeah, you just go ahead. Yeah.
Yeah. So now I will share screen. And as shown previously, UNI can develop a wide range of mechanisms to tolerate with heavy metal, and these mechanisms can be controlled entirely by phytohormone. Phytohormone, that means plant hormone, is including a group of major plant regulators like cytokinin, auxin, ABA, and interestingly, these phytohormones are not only involved in metal stress response in crop or in higher plant, they also contribute the function to algae proteins euglenoids by improve or simulation of antioxidant apparatus to minimum oxidative damage from heavy metal or stimulation of protective compound under metal stress. And in my previous review last year also show that in algae, they also have a lot of autolog for signaling plant growth regulator. For example, cytokinin, they are signaling molecule in higher plant, but the autolog of signaling molecules in cytokinin also present in algae, including histidine kinase, histidine phosphate, and response regulator. That means like cytokinin work specifically, and they need to have like signaling pathway for them, and it's present in algae. So that's why we come up with an idea to develop a new method for algal bioremediation or bioremoval of heavy metal, and to study the basic knowledge on how phytohormone was produced endogenously in algae, in euglena, and how it's respond to different metal stress. For phenotyping, I was using typical staining methods with heterocytometer and microscope to count cells under different metal stress and combine with phytohormone. You can see a screen that the dead cells always have black color, and the living cell have the survival cell have green color. And for hormone analysis, for hormonomic analysis, I divide into two. The first one, more than 50 forms of cytokinins were detected by Orbitrap, and with another phytohormone like ABA, gibberellin, auxin, salicylic acid, and germanic acid were analyzed by QTAP by different extraction method and different measurement. So, and for the hormonomic analysis, in my lab, we develop methods, and we have very good internal standards to measure the concentration of each form of cytokinin as well as another phytohormone. And why we need to care about a single form of cytokinin, because the results are absent of each form in cytokinin, and the level of them will define different phenotyping. And the knowledge from cytokinin metabolism or phytohormone metabolism contribute a lot in higher plant, for example, to develop the high yielding cultivar for soybean, for maize, or for rice. So it's very important to look at cytokinin metabolism and plant growth regulator. Now let me share the data from a study. As you can see in the slide, under control condition, ABA and cytokinin improved cell probe is not a significant difference, and under less stress, I think a concentration that reduce more than 50% of cell roll, not totally cure cell, because I want to have it itself in the end of treatment for subsequent analysis. And you can see in the slide, under less stress, ABA and cytokinin improve cell probe is not significant different also. So there are no significant effect either ABA and cytokinin on the final cell number. And for the cadmium and nickel, I also have the same condition. There are no effect of either cytokinin and ABA on the final cell number. And in this experiment, I use like medium is not induce a lot of cell roll because I want to avoid noise spectra for mass spec analysis. It is in organic media, so cell roll very slow. So that's why we cannot like see too much difference between cell roll in different concentration. And there are no effect on the cell number, and we further to see like the basic knowledge, the basic understanding, like how do cell produce phytohormone, will they produce cytokinin and ABA and how many forms of cytokinin they produce, and the level of them, and how it's respond to different metal like nickel, lead, cadmium, or when we treat it combined with ABA and cytokinin. And there are no significant effect with ABA endogenous ABA content. However, for total start cytokinin, I found that co-treated between ABA and cadmium improves significantly the total content of cytokinin, as well as co-treated of cytokinin and nickel also improve very high level of total cytokinin as compared to the control, and the metal itself also, the single metal treatment. And with isoprenoic cytokinin and achromatic cytokinin, we also found some interesting information like carbon stress increase the level of isoprenoic cytokinin as compared to the control, and the co-treated between ABA and CD or cytokinin with nickel also improve the level of isoprenoic cytokinin, and for the total achromatic cytokinin like ABA with nickel improve the level of achromatic cytokinin also. And for fraction of nucleotide is the recursor of the storage form of cytokinin, and it's very important for cytokinin metabolism. And once again, I found that ABA co-treated with cadmium improve very high fold change. I think more than 10 fold change as compared to the control. And so that it shows like ABA under metal stress, especially under nickel and cadmium, they talk specifically with cytokinin. And cytokinin with nickel also increase the high abundance of fraction nucleotide. And for fraction methyl type, based on current knowledge, we do not know too much about the function of methyl and cytokinin. They have function involved in evolution, and is one report from our study in 2017. We found that methanol also contribute for high yielding cultivar in soybean. So, and we also, interestingly, that we also found UNS cell produce some fraction of methanol under co-treated between ABA with cadmium and cytokinin with nickel. That means the application of cytokinin, the function of cytokinin and ABA under metal stress response specifically with metal, and they produce specifically response in terms of cytokinin growth bio and action. And for Thai-cysteine, cytokinin with nickel improve significant level with Thai-cysteine and Thai-IB. And when we look at another phytohormone to see possible broad talk of ABA and cytokinin with another phytohormone is very important because in higher plant system, phytohormone, they do not work alone. They cross-talk with another phytohormone and activate transcriptional regulation by recruit like general transcriptional apparatus or recruit transcriptional activation protein, increase binding or change in terms of epigenetic chain conformation of protein binding, and its effect on gene expression and finally effect on the expression of protein. So in my study, I found that ABA cross-talk with GA and cytokinin also cross-talk with GA in terms of metabolism specifically under nickel stress when increasing the level of GA1. And GA3 is very important because GA3 is the dominant form has very high content in algae and in higher plants, and they hugely contribute like in many relay functions. And we found very high level of GA3 in our sample during our study, especially when we treated with cytokinin and ABA under common stress. And there are no significant difference was found with oxygen. So the conclusion from this data, what I want to share today is that the application of phytohormone does not improve significantly the cellular number under metal stress, and the mode of action of ABA and cytokinin is specific with different metal and specific and induce specific response of cytokinin metabolism, and it's also induced the different response in terms of cross-talk metabolism with another phytohormone, for example, gibberellin. So, and in my study, I also did like when I cannot see different in terms of phenotyping, I also did like measurement of metal to see like how many percent metal uptake inside a cell, different concentration, and we found very interesting result that sometimes it improved cytokinin and ABA improve metal uptake in like different conditions, different times, and like very specific with different metal. And I also work with metabolomics to see like different pathway under control ABA and cytokinin under metal stress. For example, like metal binding peptide, it induced under cytokinin and ABA, but I don't have enough time to present it today, so maybe we will talk about this later. So in here, the table summarizes all data with the response of phytohormone under metal stress and the combined with cytokinin and ABA. And the reference I use in my study. Definitely, I want to say thank you for like all people in my lab. And today I'm very happy. I want to say thank you for your conference, so that it's a good time to me to see like the people in my lab, like my supervisor, Neil Emery, Ansley, DJ, and I also see Erin, I see Scott, and my committee member, Barry, and Emma also, like Emma worked near my lab, so I'm very happy to see you. I want to say thank you to Novogen Company and say thank you to Trent University and share the funding. So I want to say thank you for all people in my lab and Water Quality Center, DNA Building, who helped me a lot with my study. So this also led to the presentation.
Amazing. Hi, and thank you for that wonderful presentation. So now it's my turn. So I'm going to be giving a nice quick talk for you guys today as well. So I have it recorded. So good afternoon, everyone, and thank you in advance for your time and attention. My name is Emma Kaszecki, and I'm a master's student at Trent University studying under Dr. Barry Seville. And today I will be talking to you about my master's research to date, characterizing the response of a Euglena co-culture to cadmium. Canada is a leading nation in the mining industry, producing $109 billion for the GDP and providing 719,000 jobs in 2019. Despite the economic and consumer benefits, the mining industry produces an immense amount of waste in the form of mine tailings. Mine tailings are a mixture of crushed rock, fluid remnants, and precious metals left behind from the mining process. These tailings contain heavy metals and toxic chemicals and are stored directly in the environment where they remain for decades. Unfortunately, current methods for remediating areas that have been impacted by tailings are both expensive and ineffective. As a result, microalgae and eukaryotic organisms are being investigated as potential vessels for the binding and removing of heavy metals from these tailings. One of these organisms, Euglenoids, was studied by the Ngoc Nguyen group, and it was discovered that it produces metal-binding compounds known as lignin monomers and polyphenols enriched with nitrogen and sulfur. With this knowledge in mind, Victoria Kennedy pre-grew Euglenoids cultures with elevated levels of nitrogen and sulfur to assess if this would increase tolerance to cadmium. The results indicated that not only was there an increase in tolerance, but there was also an increase in the number of living cells. If metal tolerance can be enhanced in Euglenoids, can it be done with Euglenoids? Euglenoids is naturally present in acidic environments and is actually an indicator species in acid mine drainage. Research conducted by the Ngoc Nguyen group concluded that it produces the same heavy metal binding compounds that Euglenoids does. This mighty little organism is said to be a composite of 3 or 4 organisms, making its genome approximately 360 to 461 megabases. Unfortunately, though, it has not been fully sequenced or annotated. This microbe is also often present with other organisms in an apparent mutualistic relationship, thus obtaining an exenic culture of Euglenoids is incredibly difficult. However, this has led me to believe that these partnerships are essential for survival and heavy metal tolerance. Knowing all of this, I have a couple of questions. First, what are the other organisms, or partners as I like to call them, present with Euglenoids? And second, how tolerant to heavy metals is Euglenoids compared to Euglenoids? The Canadian Phycological Culture Centre provided me with 3 cultures of Euglenoids from around Ontario. Euglenoids 452 is from the Levack nickel mine in Sudbury. Along with the euglenocells, there is also a bacterial partner present in the culture. Euglenoids 657 and 658 come from a gold mine in Timmins. Along with these cells, there is also a bacteria and fungus present. We can visualize the partners present by growing the euglenocells in different environments. On the left, we have a plate of 657 grown in modified acid media with no sugar, the recommended growth media for my Euglenoids cells. This culture was grown in light, therefore it appears green because of chlorophyll production. In the middle, we see 657 grown in tryptic soy broth under light conditions, and the culture on the right is 657 grown in tryptic soy broth in the dark. At first glance, we see growth differences between these 2 types of media, and these differences are further distinguished when we examine the cells under a microscope. When grown in MAM, we predominantly see euglenocells. When grown in TSB, we see euglenocells entangled in a network of fungi. Growth of this culture with a carbon source actually enhances fungal growth to the point where the culture is taken over by the fungus. When grown in TSB in light, we see that euglenocells are present, but in less of a concentration than when grown in MAM. Euglenocells can even be seen when grown in TSB in the dark. However, at a glance, it appears that chlorophyll production is reduced compared to the light-grown culture. This reiterates the reliability these organisms have on each other and brings into question how inter-kingdom signaling is promoting the growth and cooperation of this culture. This brings me to my research objectives. First, I will assess how pre-growth with and without increased nitrogen and sulfur affect Euglenoids' ability to tolerate cadmium. Second, I will characterize transcript level changes in Euglenoids as a result of cadmium binding, and I will use these results to index against phytohormone production to identify their role in cadmium tolerance. In doing so, I will identify gene coding transcripts related to cadmium tolerance, improve genome annotation of the organism, and overall increase the understanding of how this organism tolerates cadmium. The nitrogen and sulfur pre-growth experiment was developed by my colleague Victoria Kennedy and goes as follows. 20,000 cells per milliliter of Euglenoids were inoculated into MAM, MAM with increased sulfur, and MAM with increased nitrogen. Cells were grown for 42 days, with cell counts and transfers into fresh media being completed every 7 days. After 42 days, 200,000 cells per milliliter were transferred into 0, 25, 50, and 100 micromolar concentrations of cadmium. Every 7 days, I again performed cell counts and transfers into fresh media at the varying cadmium concentrations, and each time I normalized the concentration of cells to the lowest that day. This went on for 21 days. When I would count these cells, I would first stain them with trypan blue to distinguish between living and dead cells. Cells are no longer viable when the dye is able to penetrate the cell wall and the cell turns blue. Before I show you my results, let me first outline what the data will look like.
Growth of my cells and their pre-growth media will be in the form of a line graph, with purple representing just modified acid media, orange representing increased sulfur, and blue representing increased nitrogen. Cadmium challenge cell counts will be displayed in a bar graph, with bars left to right increasing in cadmium concentrations. That is, green is 0 micromolar, yellow is 25, blue is 50, and red is 100. Here we see cell counts of each strain for 42 days in their pre-growth media. Based on these graphs, I can conclude that 657 had the greatest increase in cell counts after 42 days. I can also conclude that the increased nitrogen media resulted in the slowest cell growth, although this is most evident in 452. Overall, all 3 strains exhibit high cell growth after 42 days in their pre-growth media. Now we'll dive into the cadmium challenge data, beginning with the control graphs. It appears as though, in the absence of increased sulfur and nitrogen, all 3 strains actually exhibit quite a tolerance to cadmium. The stars indicate statistical difference in cell counts relative to the control, and this can only be seen at day 21 in 100 megamolar cadmium in 452 and across all cadmium concentrations in 658. Moving on to growth with increased sulfur, I see no difference in tolerance as a result of increased sulfur, as statistical difference can be seen at the same time point. Finally, with the nitrogen pre-growth, we again see no difference in cadmium tolerance after 21 days. I would like to mention that while there are time points in the data that make me excited for the possibility that sulfur and nitrogen have an impact, at the end of the experiment, all 3 strains in their pre-growth with sulfur and nitrogen are really no different than that of their controls. In other words, Euglena mutabilis demonstrates cadmium tolerance in the absence of sulfur and nitrogen pre-growth. A quick comparison with Euglena gracilis cells grown in cadmium shows statistical difference in cell counts in as low as 25 micromolar cadmium after only 4 days. This decrease can be seen across the entire experiment, where Euglena mutabilis was statistically different in cadmium concentrations of 100 micromolar after 21 days. This data helped me to decide to move forward with a cadmium concentration of 100 micromolar. However, I will not be pre-growing cells in sulfur and nitrogen, and therefore aim to learn about Euglena mutabilis' tolerance to cadmium in an environment similar to where it is naturally found. In order to compare molecular differences between Euglena mutabilis exposed to cadmium and its control, I need to isolate RNA for RNA-seq analysis. While the large and small ribosomal subunits for eukaryotes is approximately 3,000 and 1,500 base pairs respectively, looking at this RNA gel, it appears that in Euglena, these subunits are fragmented, resulting in the additional banding you see here. The second band on the gel, indicated by the B, was a sample of Euglena gracilis provided to me by Victoria Kennedy, and as you can see, both Euglena gracilis and mutabilis have the same fragmentation pattern. Once I have isolated RNA from my cells, it will be sent to the Center for Applied Genomics, where they will perform RNA-seq analysis. That is, they will create and sequence libraries so that I may analyze the data. I will create a de novo assembly in order to align and count transcripts I received from the TCAG. It is important to note that there is no reference genome available for Euglena mutabilis, thus the reason I need to create a de novo assembly. Through the alignment and counting of transcripts, I will be able to identify transcript level changes and specific genes associated with cadmium tolerance, and I will be able to perform blast searches to identify genes at work and confirm these results using RT-qPCR. Some final thoughts to take away from my research thus far is that a nitrogen and sulfur pre-growth do not enhance cadmium tolerance, but rather Euglena mutabilis is already quite tolerant to cadmium. In fact, it is more tolerant to cadmium than Euglena gracilis. Euglena mutabilis is also associated with other organisms. Therefore, various future analysis will be performed to increase the understanding of not only Euglena's role in cadmium tolerance, but the role of the partners as well. Gene expression differences for each organism present in the culture can be bioinformatically extracted following RNA-seq analysis. I will also conduct microbiome sequencing of this culture to identify exactly what organisms are present. Not only will this assist the bioinformatic analysis following RNA-seq, because I will already have an idea of what genes to look for based on known organisms, but primers can also be designed to identify the concentration of each organism present in the culture when exposed to cadmium through qPCR. Finally, I will conduct hormonomic analysis to evaluate the contribution of hormone signaling to metal binding efficiency using mass spectrometry to evaluate hormone responses to heavy metals. These are the references that I used for my presentation today. And I wanted to say thank you again for your time and attention.
Okay. So we're going to get into question periods with everybody now. So let's scroll up to the top. So, Saleha, you're still with us? There's a question for Saleha about what if there were any other Euglena species that you found in your research, and if you were able to determine the level of heavy metals that were present in the water systems that you were studying.
Okay, maybe the first question was like we have identified Euglena sanguinea, Euglena viridis, and Euglena gracilis. And the second question, no, in fact, we haven't determined the levels of lead, cadmium, or chromium in the water. And I think that Dr. Mahfouzur Rahman Shah has pointed out about the possibility of developing a collaborative research project. I think that it will be just great, or it will be really very good if we can develop a collaborative research project on the utilization of euglenoids by harvesting from the ponds to use them as fish or poultry feed in Bangladesh. Thank you very much for your nice idea, Dr. Mahfouzur Rahman Shah. And Dr. Sinti Bates has pointed about analysis of the biochemical composition of the biomass of euglenoids for potential use in aquafish. Yes, it's a great idea. We shall try to analyze the biochemical composition. Thank you very much. Thank you so much.
Mm-hmm. There also appears to be one more question for you, or if Sunzida wanted to jump in as well, about in the process of controlling the euglenophytes in the fish pond using lime and duckweed, was the quality and quantity of the reared fish preserved? And is there any data to compare with control experiments?
Yes, during our study, can I speak?
Go ahead, Sunzida.
Yes, during our study, we get highest production and good quality reared fish from the ponds of treatment 3, where we combinedly use duckweed and lime in our ponds. And recently we have no data about control experiment. Thank you.
Okay. And hi, I see you've already answered a question from Sergio in the chat, asking about if Euglena could be a good organism for arsenic remediation, and about phytochelidin synthase expression in Euglena and how that could improve bioaccumulation. Do you want to just touch on that quickly?
Yes. So for the first question, could you hear me now, right? This is okay, right? So for the first question, you're not good for asking about remediation. So before choosing 3 metals, nickel, lead, and cadmium for the final analysis, I tried with a wide range like 7 metals, with another one including arsenic, chromium 3, and uranium, and one more. I don't remember. So, but for another metal, it doesn't work. So that's why I picked 3 metals. And phytochelidin synthase expressions, it's very interesting to see that. Like, if I have more time, I will do that. But I did not perform like phytochelidin synthase expression. But I already performed metabolomics, like full scanning, to see different metabolite of urina produced under metal stress and in combined with fiber hormone. And interestingly, I saw a lot of peptide binding protein, like short peptide protein containing cysteine and histidine, like have ability to bind to metal. Yeah.
Thank you, Hai. It appears you touched on all the questions that were in the chat. Does anybody else have any more questions before we move on? All right. Thank you to all the speakers today. Thank God. Did you want to wrap us up then?
Yeah, thank you, Emma, and to everyone that has attended this session. So it's been very interesting, listening to the keynote speeches and also to the invited talks and then to the oral presentation. The next, tomorrow is the day 3 of the conference. We'll be basically focusing on evolution, looking at evolution of parasitism and other aspects of evolution of euglenoids. And that will be chaired by Vladimir Hampu. We would have Julius Lux, who will be talking to us, as well as other speakers. So since we have some time available, I would like to invite Scott. I think, Scott, tomorrow we have the poster session, and I think Steve might want to give a bit of an overview on how that session would work. So Scott, is Steve here to kind of?
I'm here. Yeah, sorry.
Yeah. So are you going to do this, or is it Steve?
Yeah, I thought I wasn't sure on the timing when we could do this, but Steve Barlow is our IT technical specialist here, and he's been helping to organize the spatial chat session for poster presenters. I thought it might be a good idea to field any questions that presenters have in using the platform. I've inquired with Steve if he's available now, but if you do have any questions for how to use spatial, please post them to the chat box, or we can discuss it now. Whatever your preference is, we can do both. Anybody have any comments on that?
Yeah, is Steve able to just do a very quick 5 minutes intro on how we can connect to this? Because I know he did send out a PDF, which I'm going to share with everyone today. Is he around to kind of just share his screen or something? Or?
Yep, I'm just checking with him now.
All right. Okay, while you're trying to find answers to that, I think someone just posted some comments to Emma. So Emma, do you want to go for that?
Sure. Although I have been instructed that thiols can be pretty hard to work with, I plan on moving forward with the hormonomic analysis and potentially metabolomic analysis to look and see about metal binding compounds that are present. But first using a similar approach to what Hai did, actually looking at metal uptake and hormone expression changes as a result of cadmium versus no cadmium. So not quite thiols yet, but maybe in the future.
Okay, someone is asking a question regarding poster. Is there a set schedule or an order of presentation? Okay, so I think I just posted the agenda basically for tomorrow. So that's it, Fels. In the program of event tomorrow, the poster presentation is scheduled for 17:20 GMT, so 17:20 London time. So you might just want to convert that to what time that would be. Actually, it's just about this time, but 20 minutes before now. So it's scheduled for about I think 35 minutes. So the idea behind the poster, how it's going to work, again, I wish Steve is here to kind of demonstrate, is that it will have like a screen, and then people could move from one screen to the other, then hit the button on whichever poster they're interested in, then they would have the opportunity to speak with whoever is hosting the poster there. So I don't know, I did send out a guide that was sent to me by Steve. So if you actually do go through that, that might also help. So Scott, did you manage to get hold on Steve?
I haven't been able to get a hold of Steve.
Okay, so yeah, so I think we can just have a quick tutorial about that tomorrow, basically, maybe during the break or something, or
yeah.
Just
the other thing is, if there are people that need some help, Steve is more than happy to help out, and just message now and he says that he's going to join the call, so he can come on. Literally, just message.
Oh,
so if we can wait 30 seconds and Steve will join.
Okay.
Hi, Scott. I'm here.
Oh, there he is. There you go.
Ta-da. Okay, so I understand there's some questions about spatial.
Yeah, so Steve, this is Ngoc. So we're wondering if, because Scott did tell me you got the spatials up, so we're wondering if we could do a very brief, maybe 5 minutes demonstration on how people could connect to it tomorrow. I know we have the manual, which I will send out later this evening, because I actually intentionally want to send it out like a day before, so that we don't cause some confusion. So
Sure
do you have any screen up so you can demonstrate? Or?
Yep, I can pull it up here. Are we mainly focusing on presenters or everybody?
So we can focus on everybody, and then we can then talk about the presenters later.
Okay, no problem. So I'll just share my screen here. Okay, I'll just be a second here. It's sharing within a sharing thing, so it's interesting.
So are you sharing your screen now? Or?
Yeah, I'll just be a second. I had to give permissions to Zoom. I'll just be a second.
Okay.
Okay, can everyone see my screen now?
Yeah, we can see it, Steve.
Perfect. Okay, so this is the landing spot. So everyone that clicks on the link includes the password as well for guests and presenters. Presenters have a bit of a different permission level. So you'll be dropped into this room here, and I'm appearing with my icon S, or you can, when you first log in, it'll show, actually, I should probably do that first. I'll just back up. I got ahead of myself.
So to everyone, after this meeting, I'll be sending out the login, the manual for this, so that it's just a few 1 or 2 pages to how you can log in and get to what Steve is trying to demonstrate.
Yeah. So right now, if you go to the web page, this is what will show. So basically a countdown until tomorrow. We'll go in, we'll skip. This is where you can put your name in. You can change your picture to profile picture and a little bit about yourself. Then you continue, and this is the screen that you test to make sure that your mic and your camera operate properly. So when you talk and it's given access, the green bar there will move. And you can toggle camera and microphone over on the left there. For demonstration purposes, because we're doing this through Zoom, I can't enable that right now. So basically you join the space and it'll drop you into this room. This is the main lobby, and other people will show up as icons as well. And when you move closer to them, it's basically spatial, so they'll get louder the closer you are to them, and if you move away, they'll get quieter, and you can go around and mingle that way. And then I broke up the poster sessions for each presenter over on the right here. The little icon with the person in it is a stage, so that's where the presentation will be done. So if we go in there, the presenter will need to go on stage, and they'll be granted this again. And then when I go on the stage, I'm on the stage. Now, all the people that aren't on the stage, their audio won't be heard, but you can have up to 6 presenters on stage at once. And then from here, they would go down to do share screen. And this is where they would choose the presentation. I can pull up, see if I can pull up something here.
So Steve, hopefully, Steve, will you be around tomorrow as well? I mean, during the, at 15.
Oh, of course.
Yeah. So please,
of course.
Yeah, 15, 20. Yeah. So if you can come around 17 o'clock GMT +1 and just hang around, that would be great.
Oh, for sure, no problem. So then they can go in here and do a presentation. So I'm just selecting a PDF. It can be a Google Doc, a Google slide, or anything like that. Did that not come up? That did not come up. Let's see here. Share screen. Window. So you can do the entire screen, which I don't recommend, or a Chrome tab or a window. So if it's a PowerPoint, you could share it from in here and you just click it and then it shows up next to it. Or if it's a Google slide, you can do it from here.
All right, perfect.
And then after the poster sessions, I set up breakout rooms. So basically that would be like you could go into it afterwards and have an open discussion, or you can use the chat function during the presentation up here on the right.
Okay. Okay, perfect. That sounds good. And so I suspect that the login details for the speakers, the poster presenters, should be slightly the same. I mean, it should be self-explanatory as well in the instructions.
Yeah.
All right.
Yes,
perfect.
definitely. And just a minor note, if we're jumping back from Zoom to Spatial, make sure Zoom is closed down. Otherwise, you might end up with issues of Zoom not letting go of the mic or the webcam. So it's best to close that down and then just start in Spatial.
So when closing down here means minimizing it and muting the Zoom.
Honestly, it'd be best to close it completely just to avoid any hiccups.
All right. Okay, perfect. So thank you, Steve, and I look forward to seeing you tomorrow as well.
No problem at all.
Any other question from anyone to the presenters? I'm sure this is clear. Any question, please feel free to email Steve rather. And yeah, I will see everyone tomorrow. Any comments from anyone? AOB? All right, thank you all for coming. Tomorrow we'll have a great session as well with Vladimir Hampo, Julius Lux, and many other presenters in Evolution. See you tomorrow.
Thank you.
Thank you. Bye-bye.